So next up, we have Nuvectis Pharma, Inc. It's a clinical stage biopharmaceutical company focused on the development of innovative precision medicines for the treatment of serious conditions of unmet medical need in oncology. Today we have Mr. Bentsur. Forgive me, has 20 plus years of senior leadership. He's the chairman and CEO, and his leadership experiences in the biotech industry. He served as CEO of UroGen Pharma Inc. from 2015 until 2019, and as CEO of Carex Biopharmaceuticals Inc., which was acquired by CAGLA Therapeutics from 2009 until 2015. At UroGen and Carex, Mr. Bentsur led the clinical development, regulatory approvals, and commercial infrastructure build-outs for the U.S. commercial launches of Jelmyto and Auryxia, respectively. Mr. Bentsur also led the establishment of a successful worldwide partnership for an earlier stage program at UroGen and an ex-U.S. development partnership at Auryxia and Carex. He also served as a member of the board of directors of Stemline Therapeutics, Inc. from 2009 through the approval and launch of Elzonris and the subsequent acquisition of the company by the Menarini Group in June 2020, and serves on the board of directors of Beyond Air Inc. He has an MBA from New York University Stern School of Business. So very excited to hear. Thank you.
Thank you very much for the introduction. It's a pleasure to be here. I want to thank CAGLA for inviting us to present at the conference. My first time in New Orleans, and it's fascinating. It's really a thrill to be here. So my name is Ron Bentsur. I'm the CEO of Nuvectis Pharma. I'm also one of the three co-founders, and we're focused on precision medicine for oncology. Obviously focused on areas of unmet medical need within the space. I will be making some forward-looking statements, so I do encourage anyone who's interested in learning more about Nuvectis to read the risk factors and all the disclaimers and the forward-looking statements that appear in the press releases and, of course, in the 10K. So by way of key highlights, we have two compounds in clinical development. The first one's called NXP-800. The second one is called NXP-900. The NXP-800 compound, before I go into detail about the mechanism of action and the preclinical data and things like that we've seen, the key highlights there in terms of development is that it's currently in a phase 1B program. So it already finished the phase 1A all-comers dose escalation. And in the phase 1B, we're focused on two key diseases right now. The first one is called ARID1A mutated platinum-resistant ovarian cancer. That's a very long-winded statement for a certain indication that I'll describe in a moment. We have fast-track designation for this indication that was obtained about a year ago from the FDA. And we will have a preliminary data readout from this phase 1B in a matter of a few weeks, which will include safety and preliminary efficacy. The second disease cohort that we have as part of this phase 1B umbrella for the program is in cholangiocarcinoma, cancer of the bile duct. That's a study that is being done in collaboration with the Mayo Clinic, started about 2 months ago or something like that. The second compound, NXP-900, is currently in a phase 1A dose escalation, so it's about a year behind in terms of its stage of development as compared to NXP-800. A very exciting compound. We'll talk about the merits of both in later slides. And as the gentleman alluded to, as the three co-founders of the company, collectively, we have a fair amount of experience in the industry. So you can see down below the three companies that we've been involved with, over the last 12 years or so. So I was CEO of both Carex and UroGen. I was a longstanding board member at Stemline Therapeutics. And up above, you see the names of the drugs that was approved in each one of these companies, respectively. So Auryxia, which is not for oncology, it's for dialysis patients. That was the Carex approval. And the two latter ones, Jelmyto and Elzonris, are actually for unmet medical needs in oncology. Jelmyto is for a subset within bladder cancer, and Elzonris is for a small hematological indication called BPDCN, which is kind of an offshoot of AML. We all worked together at Carex for a number of years, and then we all left right around the same time after the approval of Auryxia and the launch in the U.S. of Auryxia. I left to join UroGen as CEO. Enrique and Shay left to become heads of clinical and regulatory at Stemline, where I had been an incumbent board member. And then Jelmyto was approved at UroGen. Elzanris was approved at Stemline. Stemline got acquired, and we all kind of freed up. I left UroGen, they left Stemline, and we decided to join forces and start Nuvectis, which is what we did in 2020. Again, just to kind of recap where we are in terms of the pipeline. So the lead compound, NXP800, is in the phase 1b, which is currently comprised of 2 disease cohorts. One is this ARID1A mutated ovarian cancer, which really falls into 2 buckets within ovarian cancer. The first one is called ovarian clear cell carcinoma. The second one is called ovarian endometrial carcinoma. These are the 2 pockets where you find the ARID1A mutation. This particular study is being done in collaboration with 2 very important consortiums within the world of women's oncology, NGOT, which is the European group, and GOG, which is the US group comprised of some of the top centers in the country. And the second disease cohort that we have that is ongoing for NXP800 as part of this phase 1b is in cholangiocarcinoma, and that started about 2 months ago in collaboration with the Mayo Clinic. And NXP900, the second compound, is currently in its phase 1a dose escalation. So let me talk a little bit about NXP800 and talk about the potential catalysts. So NXP800 was in-licensed by us from the Institute of the Cancer Research in the UK, which is kind of the equivalent of the NIH or the NCI here in the US. Strong pedigree, as you can imagine. So drugs such as Zytiga for metastatic prostate cancer came from there. And the second drug that comes to mind is the recent AKT inhibitor that AstraZeneca got approved, also originated from the ICR. By way of mechanism of action, NXP800 is actually an activator of a kinase called GCN2. And what we've seen is a synthetic lethality effect with the ARID1A mutation. Those are the types of tumors that we feel can be most vulnerable or can be most affected by our drug, NXP800. So obviously that's kind of the direction that we're focused on. This is an example of some of the preclinical data that was generated. These are kind of classic ARID1A mutated ovarian clear cell models, and what you see here is a very nice effect of NXP800 in red as compared to platinum-based therapy in green. The reason we took cisplatin as a comparator here is because platinum is what's used as standard of care frontline therapy for these patients. It's not approved there, but it is used as standard of care. Generates about a 30 to 35% response rate frontline, and really nothing to speak of second line, 0 to 5%. And of course, that's where the true unmet medical need lies, and that's where we hope that NXP800 can have an effect. You can see the addressable patient populations there. So each one of these subgroups that we're focused on within ovarian clear cell carcinoma and ovarian endometrioid carcinoma, each one represents about 10% of the overall incidence of ovarian in the U.S. So the incidence of ovarian in the U.S. is about 22,000 annually. So each one of these is a little over 2,000. But keep in mind that we're looking for the patients that have the ARID1A mutation. That's the biomarker that we're using to enrich the patient population, because we think these patients should respond best to our drug. So if I do the math there, so about 70% of the patients with ovarian clear cell have the ARID1A mutation, and about 40% of the patients with ovarian endometrioid have the ARID1A mutation, for a tally of about 2,000 patients by way of incidence. By way of prevalence, it's a little over 3,000. That number may seem small, but again, for a small or mid-sized company to start off with an indication like that could be quite compelling. And another thing, just for reference, I'll mention is that the PARP inhibitors, which many of you may be familiar with, their first approvals before they got the BRCA mutation approval in breast and all that, their first approvals were in ovarian cancer and BRCA mutated ovarian. And in fact, their target patient population within ovarian was smaller than what you see here. And they were still able to generate $500-600 million within that indication. This is the data that was generated preclinically by the Mayo Clinic. These happen to be PDX models, patient-derived xenografts. You can see at the bottom right the actual results, and you can see in red NXP 800 versus the control in blue, which obviously goes through the roof because there's nothing there to stop the tumor from growing. And on the right, you actually see the pictures of the tumors for comparison. They did this about 4 or 5 times, and obviously they were very pleased by the preclinical that was generated, and that prompted the discussion for what is now the second disease cohort that we have in the phase 1b in cholangiocarcinoma, being quarterbacked by the Mayo Clinic. Another disease that we're very keen on starting as part of the phase 1b, which would be a natural extension to what we're doing in ovarian, is another women's oncology type of an indication, and this would be endometrial, using the ARID1A as a marker as well. You can see some of the data preclinically in these xenografts that's been generated. So certainly something very compelling for us to pursue. But again, we have to do things modularly. We're a small company. We can't just jump into the pool and try to cure death in every which way possible. You have to be very thoughtful and very staggered about your approach, and that's what we intend to do. So we hope to add the endometrial arm into the phase 1b within a few months. So these are the target addressable patient populations that we're talking about here. This is, again, these are kind of numbers that you can find off of the internet. These are all diseases where we've got some preclinical data. Obviously, we've got kind of in our own heads the priority of what we want to pursue and when. But you can see that there's really no shortage of opportunities. Obviously, the number of endometrial patients is really striking. As an example, this would be, a target addressable patient population by way of incidence of close to 25,000, by way of prevalence probably close to 50,000. And in terms of the clinical programs that were already conducted, that's the phase 1A dose escalation, and now the phase 1B, which is being conducted. So the phase 1A was a standard all-comers dose escalation study. We didn't have any ARID1A mutated patients within the phase 1A. But what the phase 1A did allow us to do was retrieve a lot of information regarding safety and tolerability, and of course pharmacokinetics and pharmacodynamics, and to allow us to really make an informed decision regarding the doses that were carried over into the phase 1B, which is what we're utilizing now, which is 50 milligrams and 75 milligrams once a day oral capsule. And of course, the purpose of the phase 1B is to continue to assess safety and tolerability, but also to assess safety in the specific target patient populations. And the key highlight from the phase 1B in terms of a near-term catalyst is what we expect over the next few weeks in this month, in the month of March, which would be preliminary data from the phase 1B. Obviously safety and tolerability, but also preliminary efficacy. So hopefully that'll happen within the next 2-3 weeks. With that, I'm going to shift gears and talk about our second compound in development, a drug called NXP 900. This is a compound that we in-licensed from the University of Edinburgh in Scotland. And by the way, it's just by coincidence that both of these compounds came from the UK. And what's interesting about this compound is that it's a new and improved SRC kinase inhibitor. So obviously that begs the question, why bother with a new and improved SRC kinase inhibitor? There are other SRC inhibitors out there. In fact, there are 2 that are approved, dasatinib and bosutinib, which are technically considered multi-kinase inhibitors that also inhibit SRC. And they're approved for CML and AML, 2 hematological indications, and they do quite well. Collectively, they generate about $2.5 billion. So obviously, there are no slouches in these indications. So again, it begs the question, who cares? Why bother with another SRC inhibitor? And here's why. So when you look at the history of the development of dasatinib and bosutinib, obviously they succeeded in CML and AML, but back in the day, they were tested extensively in the solid tumor setting as well, and actually showed some pretty interesting activity in certain diseases. But they always ran into major systemic safety issues in the solid tumor setting that prevented their approval in solid tumors. And so obviously the question is, why did they have these systemic safety issues? They had these systemic safety issues because they are very non-selective. They're very what's called promiscuous. They hit a whole bunch of targets. You'll see that on the next slide. So obviously in certain settings that can create problems. Too much of a good thing can cause major systemic safety issues. And that's in fact what they saw when they were treating patients in the solid tumor settings. So NXP 900, our compound, was designed from day one, from the get-go, rationally designed to overcome that by being a much more specific, much more targeted SARC inhibitor. And a picture's worth 1,000 words. So this is the kinome heat map. You can see dasatinib on the right, lights up like a Christmas tree, hits a whole bunch of things. And obviously that can be too much of a good thing in certain settings, whereas NXP 900 on the left was rationally designed to be a much more specific, much more selective SARC inhibitor. That's basically what you see on in the upper left corner. And so, they were able to achieve the development of a much more specific SARC inhibitor. Another advantage that we believe that NXP 900 can bring to bear is that we believe that it's a more potent inhibitor of the SARC kinase. How do we know this? Because if you look in the middle, for example, this is a fully active SARC kinase. And of course, in a cancer situation, this is hyperactive or overactive, and you want to shut it down. Now, just like most kinases, not all, but the overwhelming majority of kinases, this kinase has two active domains: the catalytic domain, which is where you see in the middle there, that little phosphate molecule, that little circle. That's where the game of signal transduction or hot potato takes place. At the end of this, obviously, sequence, there's a command for the cell to do something. And up above, where you see these blurbs of turquoise and brown, that's where the scaffolding domain, that's where the docking station is. Ideally, you want to shut down both the scaffolding and the catalytic domains of the kinase. On the right side, you see what dasatinib is able to do. So dasatinib is able to shut down the catalytic domain, so you don't see the phosphate molecule down here. So that's a very good start. So the signal transduction process, the messaging, the FedEx part of the equation has been stopped. But the scaffolding domain is still very much intact. How do we know that? We know that because when you look at the three-dimensional crystallography picture of the binding of dasatinib to the SARC kinase, you see that the SARC kinase remains in what's called an open conformation. It's still upright. It's still partially active. That's not an ideal situation to be in. If you go to the far left, this is what NXP 900 does. It's able to shut down both the catalytic and the scaffolding domains of the SARC kinase. And in fact, when you look at the three-dimensional crystallography picture of the binding of NXP 900 to the SARC kinase, you see that the SARC kinase is in what's called a closed conformation. It's collapsed. That's exactly what you want to see, because then you know that the kinase is completely incapacitated. So we do believe that there's potentially a mechanistic advantage as well that NXP 900 can bring to the table. In terms of the potential diseases of interest, so we're starting to focus more and more on cancers of squamous cell origin. So I don't know if many of you know this, but for example, when you look at head and neck cancer, about 80% of the cases are of squamous cell origin. Esophageal is also a very large percentage, and there are a number of others. This is some of the preclinical data, in vivo data that's been generated in these disease settings. So obviously it's very encouraging. And these are all things that ultimately, obviously, we would want to pursue in the phase 1b setting when the phase 1b starts later this year. What's particularly interesting about this compound is that we already have what we call third-party unsolicited preclinical validation for potential combination strategies. This is a very busy slide. I'm not going to bog you down with any of this data, but what you see on the left side is the highlight slide or table from a publication that was put out by the AstraZeneca R&D group about 1.5 years ago in a prestigious journal called Nature Communication. Some of you are familiar with probably. And what they did here was the following. So they've got this blockbuster drug called osimertinib, which is Tagrisso for non-small cell lung cancer. It's going to do $1 gazillion this year. Everyone's happy. But we all know that eventually patients become resistant to Tagrisso treatment, and they need to stop being treated with Tagrisso. So obviously companies such as AstraZeneca are looking for ways to overcome Tagrisso resistance. So what the researchers here did was they took Tagrisso-resistant cell lines and they tried to resensitize these cell lines with Tagrisso treatment by taking Tagrisso in combination with a bunch of other drugs. And lo and behold, our drug, referred to here as ECF506, that's the name that the University of Edinburgh, which is where we in-licensed the drug from, that's the name that they had for the compound, but it's NXP900, our drug. Our drug won this dog and pony show. And it's the only drug that's actually highlighted in this publication. And then in the supplemental piece, they talk about 5 or 6 other drugs that they tried that really didn't show anything. So obviously, when you look at something like this kind of plants a seed in our minds as to a potential combination strategy down the road. On the right side, you see kind of a similar story, this time in metastatic prostate with a drug called enzalutamide, which is Xtandi. Xtandi is the famous Medivation drug that migrated to Astellas and Pfizer and all that. And what you see here is basically the partial reversal of Xtandi resistance when you combine Xtandi with our drug, NXP900. So you've got like the orange line, which is the combination, and then you've got Xtandi alone, which obviously the slopes are very different, so that's encouraging. And what's particularly interesting about this publication is that it was done by the Memorial Sloan Kettering Group, published in JEM, again, very prestigious journal, and the lead author was Charlie Sawyer. Who's Charlie Sawyer? He is actually the inventor of Xtandi. So it's nice to kind of be able to tie the loop on that. Again, plants a seed in our minds regarding another potential combination strategy down the road. So, hopefully we can pursue all these things. These are the different, market numbers and potential addressable patient populations. And you can see that there's really no shortage of opportunities. The phase 1A, the dose escalation is ongoing now. We started about 3 months ago. We started with 20 milligrams per day, which is a very low dose. We're already done with that. We're already done with the 40 milligram dose group. We're starting the 80 milligram dose group next week. And, the study should be done within about 6 months or so. And hopefully we'll have positive things to report from the study in the second half of this year. A little bit of housekeeping. So again, we finished the third quarter of 23 with a little over $22 million of cash. We finished, you haven't seen it yet, but it's not a state secret. So the year-end number will be about $19 million. The burn is about $3 to $4 million per quarter. So we run a very tight ship. I don't know if you're familiar with burn rates across the industry, but it's about as small as you will ever see. It's a fraction of what our peer companies burn, so that gives us a little bit more leeway, a little bit more leverage. So currently, as we speak, we've got about 15, 16 months of cash on hand. And also the stock is very tightly held. So when you look at the 3 co-founders and the greater than 5% shareholders, you're up to about 2/3 ownership. And the 3 co-founders, we collectively own about, 30, 35% of the stock. And believe me, it wasn't all, sweat equity. We all have screaming wives. Why did you invest so much money in the company and all that kind of stuff? So that's already behind us, thankfully. But millions of dollars went into this thing out of our own pockets. And so we're big believers in this thing. In terms of the research coverage, these are the 3 analysts that cover us and their respective banks. Over time, obviously, you want to expand that and, kind of move up the food chain. In the past, we had kind of all the who's who covering us back at Carix and Eurogen, our prior companies, JP Morgan, Morgan Stanley, everybody. So hopefully we can, we can reach those inflection points soon. And going back to the investment highlight slide, so we talked about the 2 compounds. So the key near-term catalyst is coming up in a few weeks. That's the preliminary data from the phase 1B study for NXP800 in the ovarian program. But the second half of 24, we believe, can also be very exciting with multiple data readouts from all the programs that we're doing. So a follow-up data release from the ovarian program, a preliminary data readout from the cholangio program, that's for NXP800, and for NXP900, the readout from the phase 1A, and hopefully other things to come. So that's where I end the formal part of the presentation, and I'm not sure what the format is, if there's Q&A or... Okay, great. So if there are any questions, I'd be happy to entertain those. Thank you very much. Thank you. Hey, Eric. Thanks. So the phase 1B, obviously there's safety and tolerability, but on the efficacy side, you do want to see, responses, stable disease, those kinds of things. So I think the expectation is to see a response and, maybe a couple stable disease. And I think if we can achieve that, we'll pass the litmus test in the eyes of investors. that they're going to look at it and say, yeah, this definitely has activity. It has a real shot in this indication. What ultimately it's going to do is still too early to tell, but hopefully we can pass that kind of smell test. Is there a question? I'm sorry. Okay. Oh, all right. Thank you all very much. I really appreciate it. Thank you so much.